Presentation is about overexpressing a gene named VDE virulensin depoxidase. For that purpose, we are using gateway cloning here. So if I talk about the basic function of this gene, then whenever there is a high light stress in the plant, so it is a generation of ROS, reactive oxygen species. So to prevent from that, this enzyme converts the highlight energy in the form of heat and get it dissipated out of the plant. So our goal is to check the photosynthetic efficiency after overexpression of this gene VDE. Okay, so in this gateway cloning, there are two reactions basically, BP reaction and LR reaction. Both these reactions are based upon the homologous site-specific recombination. If you can see the sites, the particular ATTP sites, ATTL sites in the vectors of this cloning technique. So basically, what we get after this reaction is like in BP reaction we get entry clone with our insert, and in LR reaction we get expression clone with our insert. The entry vector we are using is PDonor 221, and the expression is PGWB 520. It has the 52 expression vector. It has 35 as constitutive parameter, which is continuously overexpressed. Okay, so yeah, so this let's start fitting the VDE into its destination. Okay, so first we need to just extract the DNA, then we need to design the primer. As we know that I told that is a site specific recombination, that these are sites, so we need to design the primer so that it has the complement sequence to these sites. Okay, so this is our main sequence, and this is the complementary sequence to these adapter sites. Okay, so after designing the primers, we run the PCR. Then after running PCR, we get our insert with the specific flanks in the both side of the gene of interest. And so if we talk about this reaction here, so it involves BP clonase. BP clonase has its components at excision factor and ligase. So basically what it does is it actually exchanges this toxic gene, this CCDB, with our gene of interest, and gives us an entry clone having our insert with a byproduct having CCDB gene. After that, the reaction is the LR clonase. LR clonase is also based upon the homologous site-specific recombination. They come in close proximity, and then there will be a complementation between these sides, and the insert goes inside, and the CCDB gets outside with the help of this LR clonase. And we get the expression clone with having our insert. And after validating these steps, our main motive is to put this PGWB having our VDE gene inside this uh, ectobacterium, which is a method to transform plants actually. So after validating the transformants in the plants, we can actually check the measure. We can actually measure the parameters which we wanted to measure. Okay. So yeah. So during BP reaction, actually, we are not sure whether like all the plasmids had our insert or not. So for actually, for example, if the out of 100, just 10 plasmid got recombination. So yeah, for that we need to actually amplify the plasmid having our product. Okay, so the thing is, we use competent cell for that. Competent cells are basically the cells which are treated with such a chemicals that they gain the efficiency of taking the DNA which is present in surrounding of that. So here the process is that we just take the prepared competent cells and mix our the BP reaction product inside the this competent cells. Then we incubate for 5 to 30 minutes, and then we just give a heat shock at 40 degrees Celsius for 30 to 60 seconds, and return in on the ice. Then yeah, then we add SOC medium, which is required for the growth of the bacteria, and we put it in the incubator for an hour at 225 RPM. Here the cells would replicate. Okay, so now the time is to check the transformants, because we don't know whether the BP reaction plasmid which we put in the culture, the competent cells, they actually took it or not. So yeah, for that we are using this spreading method. So this we are spreading the culture over the plate LBA. So this plate has specific concentration of antibiotic. Why antibiotic? Because the plasmid, the P donor, has resistant to antibiotic. So the kanamycin. So the cell which had this plasmid having our insert, that will survive only because had antibiotic resistance. So after selecting here, we will get the colonies here. So as you can see, yeah, now we need to validate the things. So we just need to pick up the colony, then just resuspend it in the PCR reaction, then to run the PCR, then we can just run electrophoresis. Now here we can just use different set of primers like forward vector, gene reverse, vector forward, vector reverse, gene forward, vector reverse, gene, like different set of primers. So the length will be different on the gel. Okay, that's the method how we can validate. And But actually the thing is, like, while running all the reaction, PCR and all the things, there are chances that our base pair got deleted or something. So the proper method for validation is Sanger sequencing, which we should go through. So this actually gives us the proper result. So now we need to check the overexpression, like whether our gene is expressing or not. So in case of plant, we are using the syringe filtration. We're just injecting the culture inside the plant, and now we are observing, preparing slide for the confocal microscopy, and we can actually observe the GFP. GSP, why we are observing GSP? Actually, it is present in the expression vector. This is a tag, green fluorescent protein tag. It's excited at a particular wavelength. So during, if we throw that particular wavelength on this GFP, we can actually get to know like whether it is expressing or not. So if the plant had taken this gene, or it is overexpressing, then we can actually see this GFP inside the confocal microscopy results. So that's how it is get validated. Thank you so much.